Hello, I'm Bolaji Olawale, and I'm currently a research assistant at the National Institute for Pharmaceutical Research and Development at Abuja, Nigeria. And I'm here today to present on a topic leveraging computational approaches in identifying novel HER2 positive breast cancer potential therapeutics, integrating virtual screen and molecular dynamics simulation. I would like to take this opportunity to say a big thank you to the Beyond Science Initiative for the opportunity to present at this annual conference. To begin with a brief introduction into breast cancer and cancer entirely. Breast cancer is a prevalent form of cancer that primarily affects women and is one of the most widespread types of cancer that constitutes approximately 11% of reported cancer cases worldwide. However, its impact is not confined to prevalence alone. Breast cancer also exhibits a substantial mortality rate. In 2020, about 600,000 death cases were attributed to breast cancer. Among the different variants of breast cancer that exist is HER2 positive breast cancer, which is characterized by the overamplification of the HER2 gene, which results into the overexpression of HER2 receptors. The HER2 receptor belongs to the epidermal growth factor receptor family, which are proteins that are involved in several cellular pathways, which include the cell growth, differentiation, metabolism, angiogenesis, and cell survival. The mechanism of action of this protein family is it involves several steps. The first step, which involves the dimerization of the protein, which is a case whereby HER2 or HER1 or HER3 or HER4 or whichever member of the family forms a dimer with another member of the family. In this case, HER2 can form a dimer with HER2 or EGFR or HER1 or any other member of the family. This dimerization results in the phosphorylation of the intracellular tyrosine kinase domain, which leads into the activation of the HER2 protein. The activated tyrosine kinase then binds protein, phosphorylating them and thereby activating a series of signaling pathways involved in cell growth, differentiation, and other metabolic activities. And these metabolic activities involves other proteins such as the PI3K, AKT, mTOR, RAS, MAPK, MAPKK, and its signaling pathway. Moving on. Currently, there are several treatment methods for breast cancer and other form of cancer types. We have the antibody-based drugs such as trastuzumab, pertuzumab, and the likes, which are very specific to HER2 receptors. We have cytotoxic agents. We have chemotherapeutic drugs. We have targeted therapy, which involves the like of tyrosine kinase inhibitors. We have nucleic acid-based therapies, which include the mRNAs, the siRNAs, and lastly, we have the radiation therapy, among others. Among this therapy, we have the tyrosine kinase inhibitors, which this study is focused mainly on. And this group of drugs target the tyrosine kinase domain of receptors such as HER2 and block their activities. And these example of these tyrosine kinase inhibitors include gefitinib, erlotinib, lapatinib, afatinib, neratinib, and several other tyrosine kinase inhibitors that we have that are already commercially available. For the purpose of this study, we will be focusing mainly on the use of lapatinib for the treatment of HER2 positive breast cancer. To begin with, we need to discuss the mechanism of action. Lapatinib functions in treatment of HER2 positive breast cancer by inhibiting the tyrosine kinase domain of HER2, and it does this by competing with the ATP binding site of the protein, thereby inhibiting the phosphorylating activity of HER2 protein, which inhibits the activation of downstream proteins such as the PI3K, the MAPK, and thus their following signaling pathways, which thereby inhibit the survival, proliferation, and other activity of the involved cancer cells. The lapatinib has been known for its advantage as being a very effective tyrosine kinase inhibitor, and it is known to be especially active when it is used in combination with other chemotherapeutic agents such as capecitabine, especially in cases where the patient has been formerly treated with drugs such as antibody-based drugs such as trastuzumab. However, as effective as lapatinib may be. There are limitations that need to be worked on or optimized for a better result and a better efficacy in treating HER2 positive breast cancer. And these limitations include the pre-glycoprotein inhibition and egg protein inhibition. The egg protein is a protein that is involved in the regulation of the cardiac rhythm, and the inhibition of this protein could result into cardiac arrhythmia. Another limitation is the pig glycoprotein inhibition, which could affect the assimilation or absorption of this drug in the body. So this gives rise to the reason why we conducted this study to identify better therapeutic alternatives without the limitations of lapatinib and other HER2 positive breast cancer treatment that are commercially available in the market. In this study, we used computational methods in identifying potential therapeutic alternatives for lapatinib in the treatment of HER2 positive breast cancer. We began this process with a similarity search on the PubChem database using lapatinib as the query compound, and the resulting library that was generated was screened against the HER2 protein, and the best compound from this screening was used to generate a pharmacophore model, which was also used to generate a new library of compounds that were then screened against the HER2 protein to identify the best candidate. And in this docking process, this was done in several stages. We did the SP docking, which is standard precision docking, extra precision docking using the Schrodinger Maestro software, and we then went a step forward to conduct induced field docking, which is done to take into consideration the conformational changes that occur in the binding site of the protein during the binding of the compound. Then from this, we went on a step further to calculate the free energy. This allowed us to have a better insight into the nature of the bonds that were formed and the amount of energy that has contributed into the total binding energy of each complex. Then we went a step further to predict the ADME and toxicity property, which gives us the absorption, the distribution, the metabolism, excretion, and toxicity properties of this selected compound. We get to substantiate the integrity and the structural flexibility and compactness of the complex of the selected compounds with the HER2 protein. We conducted MD simulation. So in the first stage of the docking, where we identify the compound that was used for pharmacophore modeling, we identify the compound with PubChem CID 901-96902, which are the highest binding affinity with the most favorable nature of interaction. It had salt bridges, it had hydrogen bond and halogen bond. The salt bridge are known to be the strongest form of non-covalent interaction, and hydrogen bond have also been proven to improve the stability of protein ligand complex. The visual interaction, the visualized result of the docking of compound 90196 with HER2 protein can be seen in the diagram below. And from this stage, we went on to generate the pharmacophore, and this pharmacophore was used to generate a library of 1,000 compounds from the Data Warrior software. These compounds were then screened down using the R5 Lipinski rule of 5 and screened down to about 443 compounds. And these compounds were moved on to the second stage of docking. And during the second stage of docking, which was ended with induced field docking, we were able to identify 3 suitable compounds that could be therapeutic alternatives to lapatinib, and their interaction profile can be seen here and can be seen to be superior to that of lapatinib. Lapatinib can be seen to have a binding affinity of minus 11.9 kilocalorie per mole here, while we have the other compound having far higher binding affinity, with compound 56 having a binding affinity of minus 13, compound 81 minus 13 also, compound 33 now minus 19.7, and the other two compounds having minus 12 at the same time. Compound 56, however, had a pi cation bond, which is a bond known to be resistant to solvation, resistant to pH, and significantly stronger than hydrogen bonds. It also has a single salt bridge. And if we look at the likes of compound 81 and compound 339, they can also be seen to be substantially stronger in terms of the nature of their binding interaction with HER2 protein. And the 2D interaction can be seen in the diagrams below. Moving on, we also conducted free energy calculations to gain insight into the contribution of several bonds into the final free binding energy of each complex. And in this case, we are also able to prove that 3 selected compound outperform lapatinib, looking at the contribution of their hydrogen bond and the contribution of other bonds. If we look at it, we notice that lapatinib had majority of its binding energy being contributed by weak van der Waals forces. Moving on, predicting the ADME and toxicity properties of the selected compound, we were able to identify that the 3 selected compound lacked the limiting properties that lapatinib possessed, such as the HERG inhibition. You can see they all had lower probabilities compared to lapatinib, especially compound 81 and compound 339, with all other properties being okay. We then went a step further to substantiate the structural integrity of the selected compounds. So we conducted molecular dynamic simulation for 100 nanoseconds and took properties or parameters or validating properties such as the root mean square of deviation, the root mean square of fluctuation, the radius of gyration, and number of hydrogen bonds throughout this simulation period. And through this, we were able to establish that the selected compound all outperformed the standard lapatinib in all cases, with the exception of the RMSD, where we had slightly higher fluctuations for compound 81, which could be as a result of the majority of the stabilizing bonds being salt bridges, which are known to be susceptible to solvation and pH, which are taken into consideration during molecular dynamics simulation. But regardless of this, in other cases, in the radius of gyration, the hydrogen bond and the root mean square of fluctuation, the compound outperformed lapatinib and did perfectly well. With this, we were able to identify lead compounds, the compound 56, which exhibited the most superior stability, the binding affinity, and was also predicted to have the best pharmacokinetic properties compared to lapatinib. The findings of these studies provide a solid foundation for future experimental validation, which could include in vitro, in vivo studies, which can also take it a step further into clinical trials. So future direction should focus on further optimization of compound 56, which proved to be the best compound of this set, in necessary ways to enhance its therapeutic potential and address any pharmacokinetic limitation that it might possess. With this, I come to the end of my presentation, and I would like to acknowledge the Beyond Science Initiative one time again for the grand opportunity for presenting at this conference. I would like to thank ComChem as the majority of our team members from ComChem, and I would also like to thank the Computational Molecular Biology and Drug Discovery Laboratory at LAUTECH for their technical support in providing all technical equipment needed for the success of this project. Thank you so much for listening, and have a good day.